If you own horses, work around horses, or compete in the equestrian world, there is one disease that almost everyone fears: strangles. It is one of the most contagious equine diseases in the world, and despite decades of research, outbreaks still occur in boarding barns, breeding farms, training facilities, and competition stables every single year. But now, researchers believe a new vaccine may dramatically change the way the horse industry fights this disease. The vaccine is called Strangvac, and unlike older Strangles vaccines, this one was designed using modern molecular science, targeted immunology, and years of genomic research into Streptococcus equi, the bacterium responsible for strangles. According to recent studies, the results may be some of the most promising the equine industry has seen in decades. To understand why this vaccine matters so much, we first need to understand what strangles actually does inside the horse's body. Strangles is caused by the bacterium Streptococcus equi, subspecies equi. It spreads through direct horse-to-horse contact, shared water sources, contaminated tack, grooming equipment, clothing, and even human hands. Once the bacteria enters the horse's upper respiratory tract, they rapidly begin multiplying in the lymphoid tissues around the throat and head very quickly. The immune system responds with intense inflammation. The horses usually develop a high fever first. Then comes nasal discharge, lethargy, swelling beneath the jaw, and enlarged lymph nodes that can become abscessed. In severe cases, those swollen lymph nodes become so enlarged that they partially obstruct the airway. That is where the name strangles comes from. Although many horses survive the disease, strangles is far from harmless. Some horses develop complications including pneumonia, muscle wasting, immune-mediated vasculitis known as purpura hemorrhagica, or a dangerous condition called bastard strangles, where abscesses form throughout the body. Some horses even become silent carriers. Even after outward recovery, the bacteria can remain hidden inside the guttural pouches, allowing apparently healthy horses to continue spreading the disease to others. This is one of the reasons strangles outbreaks can repeatedly reappear in the same facilities. For years, veterinarians have relied on vaccines as one possible layer of protection. But older vaccines had limitations. Traditional strangles vaccines generally fell into two categories: killed vaccines and modified live vaccines. Killed vaccines use inactivated bacteria to stimulate an immune response, while modified live vaccines use weakened forms of the organism. Both approaches had strengths, but neither was perfect. Protection levels varied. Some horses still develop disease after vaccination. Some vaccines cause swelling or adverse reactions. Modified live vaccines also raised concerns because they contained living organisms. As a result, many horse owners felt uncertain about vaccination strategies. That uncertainty is exactly what researchers hope to address with Strangvac. Unlike traditional vaccines, Strangvac is what scientists call a recombinant subunit vaccine. This means the vaccine does not contain the whole bacterium at all. Instead, researchers identified specific proteins from Streptococcus equi that are essential for infection and immune invasion. Those proteins were then produced using recombinant technology and combined it into a targeted vaccine designed to train the horse's immune system without exposing the horse to live bacteria. This is a major scientific difference. Rather than presenting the immune system with an entire organism, the vaccine presents only carefully selective antigens that are critical to the bacterium's survival. The goal is precision. Researchers selected multiple proteins for the vaccine because Streptococcus equi uses several different mechanisms to infect horses, and avoid immune destruction. Some bacterial proteins help the organism attach to tissues. Others interfere with antibodies. Some suppress immune signaling pathways. By targeting several proteins at once, scientists hope to create a broader and more effective immune response. Importantly, these proteins were selected because they are highly conserved across many strains of the bacterium world. That means the vaccine should theoretically work against a wide variety of circulating strains, not just one isolated version of the disease. But theoretical science is not enough. What truly matters is what happens during the studies. This is where Strangvac became especially interesting. One of the major studies involved controlled challenge trials in ponies. In these trials, researchers vaccinated horses using the Strangvac protocol and then deliberately exposed them to Streptococcus equi. The exposure levels used in these studies were extremely high and intentionally designed to create severe disease in unvaccinated horses. That is exactly what happened in the control group. The unvaccinated horses developed classical clinical strangles. They developed fevers, swollen lymph nodes, nasal discharge, and typical signs associated with infection. But the vaccinated horses showed dramatically different outcomes. According to the published research, approximately 94% of vaccinated ponies were protected from developing clinical strangles after experimental exposure. That number immediately caught the attention of equine veterinarians around the world because in infectious disease research, especially with a pathogen as contagious as Streptococcus equi, that level of protection is extremely significant. However, researchers also emphasized something important. The vaccine did not necessarily prevent every horse from becoming exposed to the bacteria. Instead, it prevented the bacteria from causing severe clinical disease. That distinction matters enormously. Modern vaccinations increasingly focus not only on preventing infection entirely, but also on reducing disease severity, limiting complications, and decreasing bacterial shedding. In practical terms, that means a vaccinated horse may still encounter the bacteria, but the immune system responds rapidly enough to stop severe illness from developing. The immune system response generated by Strangvac appears to be particularly sophisticated. Studies showed that vaccinated horses develop strong antibody responses both systemically and locally within the upper respiratory tract. This is extremely important because strangles begins in the upper airway. Researchers also found that antibodies generated by the vaccine were capable of neutralizing bacterial proteins involved in immune invasion. In other words, the vaccine was not simply creating antibodies randomly. It was specifically blocking some of the bacterium's key survival tools. Another important study examined the vaccine during a real-world outbreak situation. This was not a laboratory challenge model. This was an actual outbreak on a Swedish farm where horses were naturally exposed to strangles. Researchers vaccinated healthy horses during the outbreak and monitored their immune responses carefully over time. The vaccinated horses developed strong antibody responses within weeks. Most importantly, none of the vaccinated horses developed clinical strangles despite ongoing exposure within the outbreak environment. Meanwhile, disease continued among unvaccinated horses on the property. This was critical evidence because it suggested the vaccine could function not only under controlled laboratory conditions, but also during real-world disease exposure. One of the most fascinating aspects of Strangvac is its DIVA-compatibility. DIVA stands for differentiating infected from vaccinated animals. This may sound technical, but it has huge implications for disease control. Traditional vaccines can sometimes interfere with blood tests used to diagnose infection. That makes it difficult to determine whether a horse has actually been infected or is simply showing antibodies from vaccination. Strangvac was specifically designed to avoid that problem. Researchers structured the vaccine so veterinarians can still use diagnostic testing effectively in vaccinated horses. This improves outbreak surveillance and allows veterinarians to identify true carriers more accurately. For disease management, that is a major advancement. Of course, no vaccine is perfect. Researchers have been carefully not to oversell the product Strangvac. It does not completely eliminate the possibility of infection. Vaccinated horses may still encounter the bacteria. Biosecurity protocols still matter enormously. Isolation procedures, temperature monitoring, testing new arrivals, proper disinfection, and careful management practices remain essential part of strangles prevention. The vaccine is not a replacement for good management. It is an additional layer of protection. But even reducing disease severity can have enormous consequences for horse welfare and outbreak control. A severe strangles outbreak can shut down barns for weeks or months. Training schedules collapse. Movement restrictions create financial stress. Horses themselves can suffer tremendously. If vaccination can reduce the number of critically ill horses, reduce complications, and decrease bacterial spread, the impact on the equine industry could be substantial. This is why many veterinarians are watching the development of Strangvac so closely. This vaccine represents something larger than just a new pharmaceutical product. It represents a shift in how equine infectious diseases are approached. Instead of relying on older whole organism vaccines, researchers are now using molecular biology, genomics, and targeted immunology to build smarter vaccines. Vaccines designed not only to stimulate immunity, but to specifically interfere with the pathogen's ability to survive inside the host. That is modern immunology in action. In the end, Strangvac may not completely eradicate strangles, but the evidence suggests it could significantly reduce the severity and impact one of the horse world's most persistent diseases. After decades of frustration, uncertainty, and repeated outbreaks, that alone represents meaningful progress. For horse owners, veterinarians, breeders, riders, and trainers, the science behind this vaccine offers something the equine industry has needed for a very long time: evidence-based hope. And remember, I am a live human being, and I am not AI.